So students, next topic is the mechanism of blood clotting. We have seen the basic of the clotting, blood clotting. Now the mechanism how the blood gets clots. So there are 3 steps. It is a stepwise process. So these 3 steps are releasing of thromboplastin. Second one is conversion of prothrombin into thrombin, and third one is conversion of fibrinogen into the fibrins. So let's start the first step, that is releasing of thromboplastin. So releasing of thromboplastin means when tissue gets injured. So this injured tissue, I have told blood clotting occurs when there is an injury. So injured tissue synthesize the exothromboplastin and the platelets synthesize the endothromboplastin. In the function of platelets, we have seen it synthesizes the thromboplastin, and that thromboplastin helps in the clotting. So basically that thromboplastin is endothromboplastin, means which is acting inner side of the vessels, and exo is outside the vessels. Okay? So there are 2 types, that is exo and endothromboplastin. Then both these thromboplastin, mean that is exo and endo, react with the plasma proteins. We have studied the plasma. Plasma have different types of the protein, that is albumin, globulin, and the importance is the prothrombin and fibrinogen. So these both thromboplastin reacts with the plasma proteins in the presence of the calcium ions, calcium 2 plus ions, to form the prothrombinase enzymes. So these prothromboplastins react with the proteins, and after the reaction they forms the prothrombinase enzymes. The name of enzyme is thrombokinase, which initiates the thrombus formation. These enzymes inactivate the heparin. Now what is the heparin? Heparin is a natural anticoagulant which is present in our blood, which prevents the coagulation, because in intact blood vessel we does not require the coagulation. So that coagulation is prevented by the heparin, that is natural anticoagulant. So this enzyme inactivates the heparin, so that's why there is a formation of the clot at the injured tissue. So this is the first step, that is releasing of the thromboplastin from the injured tissue and the platelets at the site of the injury. Second step is the conversion of prothrombin into the thrombin. So prothrombin is, you know that the prothrombin is the protein which is present into the plasma. So this prothrombin is present in the inactive form, so it gets activated by the enzyme, that is prothrombinase, which is formed in this first step. So this prothrombinase enzyme will act on the prothrombin, and this prothrombin is inactive. After the action of prothrombinase in the presence of calcium 2 ions, that prothrombin gets converted into the active form, that is the thrombin. So thrombin is the active form of that protein. So that is the simple step, conversion of prothrombin to thrombin or into thrombin. Now the third step is the conversion of fibrinogen into the fibrin. So basically this fibrinogen is also a plasma protein. So fibrinogen is a soluble protein of the plasma. It is present in the dissolved form. It gets soluble. Okay? It gets dissolved into the plasma. So soluble form. So this fibrinogen is converted into the insoluble form, that is the fibrin. So fibrinogen is a soluble and fibrin is a insoluble. So fibrin is a fibrous protein which is a insoluble into the plasma. So it will form a clot due to the insolubility, and that reaction occurs in the presence of the thrombin, which is formed in the second step. So fibrinogen is converted into the fibrin, that is soluble fibrinogen is converted into the insoluble fibrin by the thrombin. Then next, these fibrin fibers, means these fibrin fibers which is insoluble, forms the network on the cut or on the wound. Suppose student, this is the vessel and here this vessel got injury. So what happens, this fibrin fiber form the networks at this site, and after the formation of network, what will happen? The fibrin fibers form the network on a cut or wound in which blood corpuscles get trapped, and there is a deposition of the basically the platelets, and it will form the clot, that is thrombus. So now the bleeding will be prevent. Blood cannot releases outside of the vessel. So in this way, that is the mechanism of blood clotting, and this forms the clotting of blood, or which forms the clots at the injured vessels. After clotting, a pale yellow liquid oozes from clot. It means that is called as the serum. Now there are two terms, that is blood minus corpuscles means cell. If we have taken a blood and we have removed the cells, that is RBC, WBC and platelets, such a structure will be the plasma. You know that the blood is made up of the plasma and corpuscles, that is the cells. If we have removed the cells, so remaining part only is the plasma. And from that plasma, if we have removed the protein, that is fibrinogen, that is the remaining is called as the serum. The remaining is called as the serum. So here the serum is oozes out when there is a healing or clot formation, and this serum contains the different types of the antibodies which kills or fights against the bacteria and the viruses which can infect to that wound, because there is a wound, so there is a easy entry point to the bacteria and the viruses. So thereby the serum is released, and that serum have the antibodies which kills to that foreign particles. So students, here we completed our topic, that is the blood coagulation or clotting. The blood groups, we have studied the plasma and the blood cells, that is the RBC, WBC and the platelets. Now the next topic is the blood groups. So you heard about the blood groups, you know that there is A, B, O, AB blood groups are present. So what are these actually the blood groups? So basically the antigen of blood groups is present on the surface of the RBC, which is also called as the agglutinogen. Means this antigen is also called as agglutinogen. So it is a marker which is used for the blood group. Suppose this is the RBC, so this antigen is present on the surface of the RBC, which is also called as agglutinogen, students. Antibody of blood group, antigen. Okay? Antibody of blood group, antigen, means antibody works against this antigen of the blood group, is a present into the serum, that is the plasma. We have studied what is the plasma, what is the serum, and that is called as agglutinin. So antibody is also called as agglutinin, and antigen is also called as agglutinogen. So there is a reaction between the agglutinin and agglutinogen. Now these antigens and antibodies means the antigen which is present on the surface of the RBC and the antibody which is present into the serum or plasma. These are actually the glycoproteins. These are actually the glycoproteins, means which are made up of the carbohydrates and the proteins. So this is the basic information about this, that is the antigens and antibody. Now we will study the blood group system. So we study the ABO blood group system. So in ABO blood group system, there are 4 types of the blood group, that is blood group A, blood group B, AB and O. This ABO, A, B and O blood group is discovered by a scientist, that is Landsteiner. That's why it is called as father of the blood grouping. Important that you have to remember, that is father of blood grouping is a Landsteiner. So we will study this blood groups with the help of a small chart. So there is a blood group, then in this column there are antigens on RBC, then antibodies in plasma and the donors group. Means blood group, if the person have a blood group A, it means that students the antigens on the RBC is A, that is why the blood group is called as A. And at the same time, if the blood group A person's antigens on the surface of RBC is A, then the antibodies in plasma will be the antibody B, that is anti-B. Because if here antibody A is present, if the antibody A is present, what will happen, students? There is a reaction, agglutination of the antigen antigen A and antibody A. So that's why there is a cross. It means that antigen or the blood group A have the antibody B into their plasma. So this is a naturally. And donors group means A blood group can receive the blood from the A blood group itself and the O blood group. So A can receive from the two persons, that is the person which having the blood group A and the persons which having the blood group O. Next one, blood group B, it means that the antigens on RBC is antigen B and the antibodies will be antibody A, okay, vice versa, and donors group will be B blood group and O blood groups person. Then AB blood group. AB means obviously there is a two antigens are present on the RBC, that is antigen A and B, then it is called as the AB blood group. Antibodies in the plasma. Now students, there is no antibodies in the plasma of the person which having a blood group AB. Now why? Because if one of the antibody is present, so suppose antibody B is present, so what will happen? Antigen B is present. So there will be a reaction between the antigen and antibody, that is antigen B and antibody B. So that's why there is no antibodies present naturally into the plasma of the person which is having blood group AB. So the donors group for blood group AB is itself AB, A, B and O. So that's why it can receive from all the four types of the blood groups. So that's why it is called as universal acceptor. Now how it is become a universal acceptor? Because in the plasma of AB blood group there is no antibody. So suppose the person which is having blood group A and this person donated a blood to the person which is having blood group AB, what will happen? This person have the antigen A, but in the plasma of this person, that is which is having blood group AB, does not have antibody. So there will be no reaction, there will be no agglutination of the antigen and antibody. So that's why due to absence of these antibodies, AB blood group becomes the universal acceptor, students. So this point is very important that you have to remember, that is AB blood group is a universal acceptor, and the reason is because the AB blood group does not have antibodies into their plasma. Simple. Next blood group is O. O blood group means the person have a blood group. It means that there is no any antigen on the surface of RBC. So that is why nil. So blood group is called as O. But the antibodies in the plasma of the person which is having a blood group O, that is antibody A and antibody B, both are present. Now the donors group. So this blood group O can receive blood only from the blood group O. But if you have observed into the donors group, it is present into the everywhere. So that's why blood group O is called as the universal donor. Universal donor is a blood group O. Now why it is a universal donor? Because it does not have antigen on the surface of RBC. So when the person which is having blood group O donated a blood to the AB, what will happen? No antigen. AB does not have the antibody, so it can easily accept. But suppose we are considering the B. The person B requires the blood and the person which is having blood group O donating blood to the B. What will happen? In the plasma of this person, that is which is having blood group B, there is an antibody, that is anti-A. But when this blood group O donates the blood, it does not have the antigen on the surface of RBC. So there will be no reaction because no antigen, and due to this reason the blood group O becomes the universal donor. So these are two important points that you have to remember, which blood group is a universal acceptor and which blood group is a universal donor. Clear? So students, here we completed the ABO blood group. Now there is another factor, you heard about it, that is the positive and the negative. Some persons have blood group that is the A positive, B positive, AB positive, O positive, or maybe negative, means A negative, B negative. So that positive and negative is depend on the another antigen, that is the RH factor, that is the RH factor. So next topic is the RH factor. RH factor is discovered by the scientists Landsteiner and Wiener in a rhesus monkey, and that is why it is called as RH for a rhesus monkey. So that is why it is called as RH factor, and this is also called as the D antigen. D antigen. Okay students. Next, RH antigen is a dominant gene. Okay, RH antigen having is a dominant gene. So antigen, if the antigen is present, then it is called as RH positive. Antigen present means RH antigen is present on the surface of RBC, then it is called as RH positive. If the antigen RH antigen is absent, then it is called as RH negative. So in India, percentage ratio of the RH is 97% peoples are RH positive and 3% peoples are RH negative. So the negative proportion is very less, that is only 3% have the RH negative blood group. Then in the world, if we consider the world, 80% peoples are with a RH positive and only 20% peoples are with a RH negative, students. So that is why the negative blood group is a rare, because the population of negative is very less. Now in RH positive and RH negative, antibody is absent, means this for this antigen, that is antigen RH, there is no antibody for these antigens, students. So this point also important. Now the actually the O minus is a universal donor. O minus is a universal donor. Now why O minus is a universal donor or O negative? O minus means the O negative, because if the O positive, the person have a O positive blood group, it means that it does not have the AB antigen, but it have the RH antigen. And if the blood is given to the another person, what will happen, students? There is a possibility of formation of antibodies against that person. So I will explain this. Suppose there are two persons, that is the A person and B person. Okay students. Now it is a RH positive. It is a RH positive. Suppose O positive. O positive. And if this person donated a blood to the B, what will happen? There is no AB antibody, antigens, but D antigen is present. So what will happen? This B person also does not have antibody, because antibody is absent for this antigen. But when this blood is received for the first time, it is a condition, okay? That is the first time. If the same blood is a transfused, this is the second time, this is the first time. So if the same blood is transfused during to second time, what will happen? Once we have transfused the blood, donated the blood to this person, so there is a possibility of formation of antibody against this D antigen into this B. So what will happen during the second time? During the second time, when we donated the blood, the D antigen is present or RH antigen is present, and this person's body have already produced the antibodies for this D antigen or RH antigen. So there will be, what will happen? There will be the clumping of RBCs during the second time, means there will be formation of the clots, the RBCs will be destroyed. So that's why we have to say specifically O minus, O negative is a universal donor, because it does not have any antigen, not AB and not D antigen or RH antigen. Then AB positive is a universal acceptor. Okay? Why AB positive universal acceptor? Because AB positive means, it means that there is a no antibodies. There is a no antibodies into the AB blood groups. So another one term, that is the erythroblastosis fetalis. Now what is the erythroblastosis fetalis? So suppose students, if a mother having Rh negative blood and father having Rh positive blood, so there may be a chances of Rh positive blood group of the fetus or their baby. So in that condition, first baby will be normal. There is no any problem with the first baby or first pregnancy. But during the second pregnancy in this couple, if the fetus have Rh positive blood group, what will happen? There will be a death of the baby. Rh positive means that fetus have the Rh antigen. Now why there is a death of that baby during the second pregnancy? What happens, students? During the first pregnancy, some amount of blood reaches into the mother after the pregnancy and from the fetus. So if the fetus have Rh positive blood group during the first pregnancy, what will happen? Some amount of blood from the fetus enters into the mother during the delivery, and once the blood enters into the mother's body of the fetus which having Rh positive antigen, the mother will form the antibody against that Rh antigen and destroy these RBCs of the fetus. Now what happened? During this first delivery, there is already formation of the antibodies for the Rh antigen, and when there is a second pregnancy, there will be antibodies is already present into the mother. So the blood which is supplied to the fetus by the mother, which is already having the antibody for the Rh antigen, so it will kills the RBCs of the fetus, and if the RBCs of the fetus are killed, there will be the death of that baby in the early stage. So for the prevention, because there may be a chances mother having Rh negative and father have a Rh positive, so for this purpose, within 72 hours of the first pregnancy, within 72 hours, the anti-D injection is given to the mother to kill, or the antibodies, these are the actually antibodies. Anti-D injection means which having a antibodies for the Rh antigen, so is given to the mother. So what will happen? Those RBCs are given reach into the mother's blood, the antigen is killed by this anti-D. So there will be no naturally formation of antibodies into the mother. So during the second pregnancy, there is no chances of the reactions of the antigen and antibody. So that is the importance of these blood groups. So that's why we have to study these blood groups for the medical education. So erythroblastosis fetalis means there is a death of the baby which is occurring in this condition if the mother having the Rh negative blood group and father having the Rh positive blood group. So students, here we completed our topic, that is the blood groups.